Now, again from Canada, University of Manitoba, we'll have Dr. Onwah, and he'll give us a talk about the role of dihydrolipoyl dehydrogenase in the immunopathogenesis of Leishmania major. Now you can share, please. You are welcome. Unmute yourself, please.
Okay,
yes.
Thank you so much. I will share my screen right away.
Thank you. We can hear you. And you can see, yes, you can see. Yes, go ahead. Thank you.
Okay. Okay, thank you so much. Hello, everyone. My name is Stella. I'm a PhD student in the Department of Immunology at the University of Manitoba. I'd like to thank the organizers for inviting me to give the talk titled The Role of Dihydrolipoyl Dehydrogenase, DLD, in the Immunopathogenesis of Leishmania major. Cutaneous leishmaniasis is caused by numerous Leishmania species, including Leishmania major. It presents as a self-healing lesion that could be localized or diffused. Other chronic infections include progressive ulcers and the secondary disease called the recidivans, or mucosal leishmaniasis that affects the mucosae. About 1 million new cases occur annually, and it's more in countries in South America, parts of North Africa, and Asia. Leishmania has a digenetic life cycle that alters between two life forms: the flagellated promastigotes in the sandfly host and the aflagellated amastigotes in the mammalian host. The life cycle begins when infected sandfly transmits the promastigotes to the mammalian host, and these promastigotes infect various phagocytic cells. These promastigotes get transformed to amastigotes, where they extensively replicate, and after a while they get released out of the cell and either infect other cells or get taken up in another blood meal by another sandfly host, where they get converted back to promastigotes. Dendritic cells and macrophages play key roles in the functional outcome of the immune response to cutaneous leishmaniasis. Infected disease would release IL-12 to activate Th1 cells to produce interferon gamma, which activates infected macrophages by the upregulation of inducible nitric oxide synthase, which enables the production of nitric oxide that promotes parasite killing. IL-4 from different cell types favors the induction of Th2 cell development. These cells produce cytokines that inactivates infected macrophages by the upregulation of arginase. By acting on L-arginine to produce polyamines, and this promotes parasite survival. Tregs and infected macrophages also promote parasite survival. There is no approved vaccine against human leishmaniasis. Vaccination is possible because people that recover from the disease are immune to the infection. For a while, our lab has been interested in understanding the antigens that drive protection in cutaneous leishmaniasis. And we have shown that peptides derived from Leishmania's dihydrolipoyl dehydrogenase, DLD, elicit strong CD4 T cell responses, and the recombinant DLD induce protective immune responses. Now we have come to understand that DLD is a critical mitochondrial enzyme in eukaryotic cells, including Leishmania, and they are known to modulate metabolic activities. DLD in different pathogenic organisms are promising targets for therapeutic strategies. The gap in knowledge that my study hopes to fill is the role of DLD in L major immunopathogenesis. And so I hypothesized that DLD is a virulence factor and its deficiency in Leishmania major will result in an attenuated disease pathology and altered host immune response. In order to achieve these objectives, I deleted DLD in Leishmania major and functionally validated this deletion in arsenic culture in macrophages and in mice. The deletion strategy I used is the CRISPR-Cas9 system, and if you're less familiar with the technology, it makes use of guide RNA that is complementary to the gene of interest. Once it's expressed alongside a Cas9 protein, a ribonucleotide complex is formed, and this initiates a cleavage. Once a cleavage has been initiated, I induced a homology-directed repair mechanism whereby I introduced a bleomycin gene resistance cassette that contains homology arms of the cleavage site. And this is important because we want to eliminate the error mechanisms that are associated with other repair processes. Once the DLD mutant clones have been confirmed, I confirmed DLD deletion by PCR using DLD-specific primers. I confirmed DLD deletion by PCR by the absence of DLD-specific band in the DLD knockout parasite, which was present in the wild-type parasites. I made further confirmations of DLD deletion by RT-qPCR, and you can see that DLD has been deleted by the absence of DLD mRNA expression in the DLD knockout parasite, which is present in the wild-type parasites. I also confirmed DLD deletion in vivo by infecting mice with wild-type or DLD knockout parasites in the food pad, and after 5 weeks, I assessed the spleen for the presence of DLD-specific CD4 T cells by flow cytometry using DLD tetramer. And you can see that in the DLD knockout group, there was no detection of DLD-specific CD4 T cells compared to the wild-type group, which suggests that DLD has been deleted. I functionally validated DLD deletion in arsenic culture by growing equal numbers of wild-type and DLD knockout parasites over time. And you can see that DLD knockout parasite has a significant reduction in the proliferation compared to the wild-type parasites. I also wanted to see whether these parasites were compromised in macrophages because they are the permissive host for amastigot replication. And so I infected bone marrow-derived macrophages with wild-type or DLD knockout parasites and tracked infection in giemsa stain over a microscope. And you can see that in cells infected with DLD knockout parasites, they had reduced proliferation over time compared to the wild-type groups. The DLD knockout parasites in these cells also look quite different from those of the wild-type group. Statistically, there was a significant reduction in the parasite per 100 macrophages, as well as the percentage infection rate of the wild-type groups compared to the knockout group compared to the wild-type group. Now, this reduction is not because the DLD knockout parasite wasn't entering the cells. As you can see, at early time points, there was no significant difference in both the parasite number or the infection rate. This suggests that DLD is a critical mitochondrial enzyme responsible for the proliferation and survival of Leishmania major in macrophages. So since we see that DLD knockout parasites were compromised in vitro, I wanted to know whether this impaired phenotype can be observed in mice. And so I infected the foot pad of mice with wild-type and DLD knockout parasites and assessed pathology by monitoring lesion development every week. I also assessed parasite burden at 5 weeks post-infection in the foot pad of the animals. And so you can see here that in the DLD knockout group, there was no observable lesion that developed compared to the wild-type group. There was a significant reduction in the parasite burden of the DLD knockout group compared to the wild-type group. And so this suggests that DLD is a virulence factor in Leishmania major. Since I did not observe any pathology in the mice that are infected with DLD knockout parasites, I wanted to know how these parasites affect host immune response. So in the same experiment, I assessed the immune response in both the spleen and draining lymph node of the infected animals by flow cytometry and ELISA. So in the spleen, you can see a significant reduction in the levels of cytokine-producing CD4 positive T cells in the DLD knockout groups compared to the wild-type groups. However, there was no significant difference in the cell counts in the spleen among both groups. These spleen cells were re-stimulated with soluble Leishmania antigen, and the levels of cytokine produced in the culture supernatant were assessed after 3 days by ELISA. And you can see again that there was a significant reduction in the cytokine levels produced from the DLD knockout groups compared to the wild-type groups. We observed similar results in the draining lymph node of the infected animals, although in the cell counts of the draining lymph node, we see that there was a significant reduction in the cell counts of the DLD knockout group compared to the wild-type group, suggesting that in the animals that were infected with DLD knockout parasites, there was reduced inflammation going on around the site of infection. Taken together, this suggests that mice infected with DLD knockout Leishmania major developed an altered immune response. In conclusion, I've been able to show that DLD knockout parasite displayed impaired proliferation in axenic culture and in macrophages, suggesting that DLD is critical for the intracellular survival of Leishmania. I've also shown that DLD knockout parasite does not induce pathologies in mice and showed an altered host immune response, suggesting that they could be potential life-attenuated vaccine candidates. I'd like to acknowledge everyone who has made the smooth progression and transition of my project successful. I would also like to thank my supervisor for his continuous mentorship, Dr. Uzonna, and direction for my research. So thank you, everyone. I'm happy to take your questions.
Thank you.
That was a great talk, Stella. I'm going to jump in here with a question. And I also want to say a shout out to Dr. Thomas Maruka at your university, because he was a former graduate student of mine. So I'm delighted he's now on your committee. Say hi to him from me. So Stella, that's really interesting. Now, Leishmania obviously isn't a serious problem in Canada. So it's intriguing that you're doing these studies, but I guess it has relevance to the African continent, and I'm delighted that you're studying this. So you mentioned about developing a non-live vaccine with this particular knockout. What kind of a vaccine strategy would you use, given what we're learning with RNA vaccines versus protein vaccines? What would you do?
If I was going to be using my DLD knockout parasite vaccine, I'm going to be using, first of all, I have to check, first of all, that there is no pathology that is being observed at later time points of infection, because we only have just observed that there was no pathology at 5 weeks, as at 5 weeks. So we're hoping that it doesn't develop any pathology going forward. However, the vaccine strategy, I'll be infecting, I'll be re-challenging animals with, or let me say mice. I'll be re-challenging mice with I'll be infecting or inoculating this vaccine, this DLD knockout parasite in mice that will be re-challenged with live parasites. If that makes sense. So
you're going to use the whole
vaccine, the whole parasite, DLD knockout parasites.
Why wouldn't you just use a mutated form, say, of DLD, or you've shown it's a virulence factor, DLD. So if you you wouldn't try and use some sort of mutated DLD to see if you could competitively inhibit or something?
Okay, so something about what drives protection in leishmaniasis is the fact that you have to have, it has been known or it's been shown that you have to have that consistent presence of parasites to drive memory responses. So, so far, we have not seen protection from recombinant protein over time, as the presence of parasites isn't there to sustain that memory responses. So a live attenuated vaccine is a potential candidate to ensure that continued protection or memory response generated.
Great, thank you.
Maybe you can put part of the parasite and the DLD mutated one into nanovesicles, like nanovacine, sorry, nanovesicle, something like exosomes, as occurring nowadays.
Oh,
Okay.
I see. Yeah, that'd probably be a consideration.
This will be great for Africa, for traveling to Africa, and these problems like malaria or something like this.
Yeah.
Okay, we have one question. Any human clinical trials started? Clinical trials?
No, we haven't moved into clinical trials, and I'm not sure if I would be involved in a clinical trial yet for now.
No.
So we are just at the starting phase of the research.
Okay. Attendees saying great job, great presentation. Do you think this finding will hold true for other Leishmania parasite species that are either cutaneous or vesicular?
So I'm not sure if I would, okay, how do I answer this? Okay, so we are hopeful that DLD, knockout L major parasites, will be able to be used as a vaccine candidate across all forms of Leishmania infection, like the visceral, the mucosal, and all forms of it. Why we are hoping that is because we did a search and we saw that DLD has more than 90% similarity across all species of Leishmania. So we are hoping that it will be able to be used as a protective vaccine against all the forms of Leishmaniasis.
Yes, very nice. And you can make try to have a molecular docking or something like this to find inhibitors. To the protein,
if we have inhibitors?
To find a treatment.
Oh, yeah.
Immunomodulators or immunoinhibitors or something like this. Okay. Okay, thank you. We have to end questions now.